Hi, welcome back to chapter 4. This week we'll be looking at life's home, the cell. In this chapter, we will cover cells as life's fundamental unit. We'll look at two different types of cells called prokaryotic and eukaryotic cells. We'll go into detail about each of those cells. For the eukaryotic cell, we'll look at a tour of the animal cell's protein production path. And so if you remember the DNA inside each of your cells, its job is to produce proteins, and those proteins then go on to sort of coordinate everything else going on in your body. And so we'll look at how those proteins are produced, and then we will look at cell structures outside the protein production path, so what other cell structures do for the cell. We'll look at the cytoskeleton, so kind of what's giving excuse me, your cell its structure, and then we will take a look at the plant cell and how cells communicate with each other. Every life form that we know of on Earth is composed of one or more cells. Viruses are a possible exception. I'll bring up just so you know this as you hear the different terms. Viruses possess many characteristics of life, but they can't reproduce outside of their host. So they can live and reproduce inside, say, your body, but they can't reproduce out on their own, just out in the air. And they are not made of cells. So there's sort of a gray area between whether we consider them a living thing or not. They're the only thing that we know of that fits in this category, but every other living thing we know of is made up of one or more cells. And all of these cells have four characteristics in common, whether you're looking at what they call a prokaryotic cell or a bacteria cell, this one here, or a eukaryotic cell, things like plant cells, animal cells. Any of those types of cells that you look at all have a cell membrane surrounding them. So that's that sort of purplish area on the outside of the cell, and this protects the cell and controls what moves in and out. All cells also have a fluid inside of them called the cytosol. It's just all this pink area here and all the pink area here, and it sort of holds nutrients and everything else in place in the cell. All cells have ribosomes, which are structures that are involved in producing proteins, and all cells have DNA. And this, you can see the DNA is in this area here in this cell, and it's sort of all these blue lines here in this cell. And DNA is also involved in producing proteins. So those are four things that no matter what type of cell you're looking at, they will all have those four characteristics: cell membrane, cytosol fluid on the inside, ribosomes, and DNA. Now, a long time ago, obviously people did not know cells existed. So they knew about things that they could see, but they didn't know what they were made up of. And the first person to actually see a cell was Robert Hooke in the 1600s. And he was primarily looking at small organisms under the microscope, like the flea there. But he looked at cork cells one day and noticed these tiny little compartments and coined the term cell. So he called them cell, meaning like a little square compartment. And that term stuck, so we continued calling these small compartments within living things cells. This was a picture of his microscope here that he used. It was a compound microscope like the microscope you had, in that it had two different lenses in it, and these two different lenses helped magnify the object he was looking at, so that he could see all these parts more clearly. Now, his work led to a lot of other scientists wanting to look under the microscope and see what smaller things they could see under the microscope as well. One of these people was Anton von Leeuwenhoek, and he was a Dutch person and had no formal training in any higher education, especially science. But he's thought to have been inspired after seeing a drawing of Hooke's, and Hooke's had a book called Micrographia, and he saw all these little things that he saw under the microscope and drew really great pictures of, and inspired him to want to try to make microscopes that he could see through more clearly. And so he spent countless hours grinding lenses in an attempt to more clearly see this tiny world, and he made hundreds of different types of microscopes. This is one of the microscopes that you can see here, which looks a lot more simple than the other microscope, and in some ways it is more simple, that it contained a tiny little just one lens, and you would hold this thing up to the light and look through that tiny little lens at the object. So instead of being magnified by two lenses, it was a single lens. But he got so good at grinding these lenses that they think that some of his microscopes could magnify up to 500 times. And if you'll notice your microscope that you have at home, if you were to use the highest power lens, which we don't even use in this class, it could magnify up to 100 times. But his microscopes back then, just due to his excellent grinding of the lenses, they thought could magnify about up to 500 times. And so by seeing things this magnified, he was able to discover an entirely unknown world of tiny things such as giardia, bacteria, sperm cells, plant spores, and he called them animalcules, meaning little animals, and all these things that he saw living within other living things to him look like tiny little animals. Here's a picture of some of the things that he saw. So when he was looking in pepper water, he could see some of these little seeds here and what the features on those seeds look like. This is what he saw looking in hay water. These were some different sperm that he saw from the human, from a ram, from a frog, dog, rabbit. And so he drew all those and compared those. And he called this an itch animal here. And so a mite is here. He was able to see all these different things living amongst the world that currently at that time they had no idea existed. So he really opened up scientists from that point on to realizing that there was a lot more out there than we previously knew about. And I thought this was an interesting quote from Anton von Leeuwenhoek, that he said, "My work, which I've done for a long time, was not pursued in order to gain praise I now enjoy, but chiefly from a craving after knowledge, which I notice resides in me more than in most men." And Einstein made similar comments to this, and that he really wasn't that smart, although I think he probably was very smart, but it was just this craving for knowledge and this curiosity that really led him to discover these new things. And I think that's great to keep in mind, that regardless of our past knowledge or where we're at, that it's that drive to want to pursue something more and want to learn about things that really brings out knowledge in any of us, and especially in the scientists who really pursue that. So I talked a little bit about a prokaryotic cell, the bacteria, versus eukaryotic cells such as plants and animals. Let's talk about some things what those cells are and what features those cells have. So on the left-hand side here, we have information about eukaryotic cells, and the word eukaryote comes from the Greek words eu, which means true, and the word karyon, which means nucleus. So the word eukaryote in Greek means true nucleus. And what this means is that these cells have their DNA contained within a compartment with a membrane called a nucleus. So this nucleus sits here in the center of the cell, contains the DNA, and it's surrounded by a membrane holding that DNA in place. If you were to look at a prokaryotic cell, or these bacteria cells, this comes from the Greek word pro, meaning before, and again, karyon, meaning nucleus, so it means before nucleus. So these were cells that did not have their DNA contained in a nucleus, or there's no membrane surrounding that DNA. And so what this means is the DNA just floats around loose inside the cell. Eukaryotic cells are usually made up of multiple cells. There are a few that are made up of one cells, but in general, like your own body, can contain trillions of cells in a eukaryotic organism. Prokaryotes, or bacteria, are always made up of only one cell. Prokaryotic cells contain only one type of organelle for producing proteins. These are the ribosomes, and they're relatively small compared to a eukaryotic cell. And eukaryotic cells contain lots of organelles, or little organs, and these are in a eukaryotic cell, these are tiny little structures that are capable of doing different jobs, such as producing energy or cleaning out waste. And so it has all these compartments within the cell that do different jobs. And again, they're relatively large compared to a bacterial cell. So still very small compared to probably what we see on a daily basis, but larger than a bacterial cell. So again, eukaryotic cells means true nucleus. Their DNA is contained in a nucleus. These organisms are usually made up of multiple cells, and they contain lots of different organelles doing different jobs and are relatively large. Prokaryote means before a nucleus, their DNA is loose, not contained in a nuclear membrane. They're always made up of only one cell, and their cells contain only one type of organelle for producing proteins and relatively small. So let's go into the parts of a eukaryotic cell. And whether you're looking at a plant cell or an animal cell, these cells all have some common features. And one is that they have a nucleus somewhere in the center of that cell that holds the DNA. They have lots of different organelles or different structures, and you can see those colored in this picture outside the nucleus, but still inside the cell, that do different jobs. We'll go into all those later, what jobs they do. There's the fluid, this blue here, called the cytosol, that holds everything in the cell, holds it in place. It also holds nutrients and support structures. And when you hear the term cytosol, that's referring specifically to the fluid inside there. If you hear the term cytoplasm, that means the fluid plus everything floating in the fluid, such as the nutrients and support structures. All eukaryotic cells also have a cytoskeleton. You can see here these colored sort of rods, and they're protein fibers found inside the cytoplasm that help give the cell its shape. It's similar to how your skeleton, your bones, give you shape. Cells have the same thing. They have all these structures that help give it shape and make it be the nice round shape that it is instead of just being flat, smashed. And lastly, all eukaryotic cells have a plasma membrane or a cell membrane. That's the outer lining of the cell. Let's start by looking at one type of eukaryotic cell, again, the cell with a nucleus, and that's the animal cell. And then later we'll look at what the plant cell looks like in comparison. So animal cells are round. You'll see later that plant cells are square, like the quark cells that Robert Hooke saw. Animal cells are surrounded to and connected by cells of a similar type. So for example, your skin cells are surrounded by and connected to other skin cells that are very similar. Blood cells are surrounded by other blood cells that are very similar. The animal cell is about 25 micrometers. We use this symbol here, micro and then meter, in diameter, which means this distance from here to here is about 25 micrometers. Now, micrometer is 1/1,000,000th of a meter. And just to give you an idea of size in a little more practical terms, if you lined up these animal cells end to end, you could fit 30 of them in a dot the size of this period. So pretty small, but not nearly as small as the bacterial cell. Now let's look at next what goes on in an animal cell. And we've talked a bit about how DNA is the main storage of information telling your cell what to do. And the way that it does that is producing proteins, which then each have a job that they do. And so we're going to go through that process of how we get from DNA to getting a protein. And so here we have, this is the nucleus of the cell where your DNA is held. And so your DNA here in the nucleus gets first copied, and then that copy gets sent out of the nucleus. It's used through a long process to make proteins. Those proteins get packaged up and then shipped out of the cell. And we'll go into each of those steps in a little bit more detail next. But just to give you a general idea, cells are a lot like a factory. They put together a product, which is which are proteins along a sort of assembly line. They package that product, those proteins, and then they ship it out of the cell. Let's start breaking down this process of how proteins get made. We're going to look at this first step here of where we take our DNA and copy it. And this all happens inside the nucleus of the cell. So here's your nucleus here, looking at a little bit bigger picture of the nucleus here. Inside your nucleus, you have all this DNA floating around. And remember, DNA is like a recipe for making up proteins. The recipe will call for different amino acids, which are the building blocks of proteins, to be made and assembled. And the order that they're assembled in determines the type of protein you get and the job that protein will do. But the proteins don't actually get made in the nucleus, and the DNA does not ever need the nucleus. That DNA does not ever leave the nucleus. The DNA is super important. We don't want it to get compromised, or everything in the cell would not work, and everything in that living organism wouldn't work. So there's a clever way that this gets done, the DNA, instead of ever leaving the nucleus or getting messed with, gets copied onto something called messenger RNA. And we abbreviate that mRNA. And what this mRNA does is it takes one strand of the DNA and it makes a copy of it. And so mRNA is a one-stranded copy of what's on the DNA code. And again, DNA is two strands. We have these two spiraling strands. mRNA is simply one strand, but then that mRNA has the ability to leave the cell through what they call a nuclear pore. These are just holes or openings in the nuclear membrane, or that membrane surrounding the nucleus. And your DNA is not allowed to leave the nucleus, but mRNA is allowed to leave through these pores. So remember, the DNA gets copied onto this messenger, really like DNA, but only one strand. They call it messenger RNA, and that's what gets to leave the nucleus. So, and that's just so that your DNA doesn't have to leave the nucleus. It can stay there and stay as that set of instructions inside your nucleus. So this messenger RNA leaves the nucleus and it travels to a ribosome. And this messenger RNA goes through the ribosome and it tells the ribosome, it's a code that tells the ribosome what amino acids to start producing. And if you remember from last week, a protein is a chain of amino acids. So this chain of amino acids starts being released from the ribosome. And the amino acid chain will start to fold up, and in the order, it will start to fold up in the way that's determined by this code on the messenger RNA. And so it will start to fold up and become a very short, but not yet fully functional protein. So the protein chains made up at this point are short, they're not fully functional, they need other things in the cell to finish being made. Once ribosomes start reading this stretch of messenger RNA, they produce, as I mentioned, this short amino acid chain. And that amino acid chain will tell them to do one of two things. It will either tell them to go hook onto this structure called the rough endoplasmic reticulum, or it will tell them to stay in the cytoplasm of the cell. So let's look at next what happens when a ribosome decides to hook on to this endoplasmic reticulum. So let's just break down that word first. Endoplasmic reticulum. The word endo means inside, plasmic means cytoplasm, and reticulum means network. So when we put all that together, endoplasmic reticulum means network inside the cytoplasm. We call it the rough endoplasmic reticulum, or we can abbreviate it RER to shorten that down. It looks rough or bumpy due to all the ribosomes that are located on it, so it has a bumpy appearance. Ribosomes hook onto the rough endoplasmic reticulum once they've started to make this short protein chain or amino acid chain, and they hook onto the endoplasmic reticulum, they drop off these unfinished proteins into the chambers within the endoplasmic reticulum, and a chain of sugar is then added to it. The rough endoplasmic reticulum will do some quality control, break down any pieces that don't look good, that weren't made correctly, and get rid of those. And then decide the ones that do look good to package those up and send them on their way. So your cells are pretty amazing that inside your cells, all of these things are happening. They're able to look at what's good and what's not good and decide what should be passed on to the rest of your body and what shouldn't be passed on to the rest of your body. Now, so that's what happens to ribosomes that decide to hook onto the endoplasmic reticulum. But a lot of the ribosomes, you can see in this picture here of this animal cell, these little blue dots are just the freely floating ribosomes. You can kind of see some of them here too. And so some of them decide not to hook onto the endoplasmic reticulum, and those specific ribosomes will produce proteins that will be used within the cell's cytoplasm in this area here, or produce proteins that are needed for the nucleus. So they produce proteins that stay within the cell. Things that the ribosomes that hook onto the endoplasmic reticulum, for the most part, are producing proteins that are going to be exported from your cell and leave the cell. So we've talked a lot about the importance of ribosome to produce the proteins that control everything else. Let's talk about now where those ribosomes come from. And where they come from is something called the nucleolus, which is an area within the nucleus. So if you look at the nucleus of your cell, there's this ball inside the nucleus. It looks almost like a nucleus inside of a nucleus. And this is called the nucleolus. And the nucleolus produces a special type of RNA, so another type of copy of the DNA, and we call this ribosomal RNA or rRNA. And that's this right here. And it begins to build a ribosome. It doesn't finish the ribosome in the nucleus or the nucleolus. It produces this unfinished ribosome that will then leave the nucleus, go out into the rest of the cell, and then finish being built out in the rest of the cell. So it needs some things from out in the rest of the cell to finish being built. As I mentioned earlier, ribosomes start producing the small protein segment, and that tells them either to attach to the rough endoplasmic reticulum, as we talked about, to produce proteins either for the cell membrane itself, so that outside layer of the cell, or to be exported outside the cell. If the ribosomes stay floating, though, they'll produce proteins for the nucleus or the cytoplasm. So again, ribosomes are start being made within the nucleolus of the nucleus. So way down here in the center of the cell is where ribosomes start being made. And they are made with this special type of RNA or special copy of your DNA called ribosomal RNA. Once ribosomes produce a protein, they need a way to be transported to where they are going. And the rough endoplasmic reticulum, or the RER, is basically just a large folded membrane, and pieces of this membrane can fold around a protein that's made. They can bud off, sort of make like a bubble or a sac around the protein, and then these sacs can carry the protein safely to wherever the next destination is that they need to go. A lot of the proteins will go to something called the Golgi complex, and this is where protein sorting and distribution occurs. So in our overview picture here, this is the Golgi complex. So these proteins leave the endoplasmic reticulums in these little sacs. They go up to the Golgi complex. The protein gets dropped off fuses with the Golgi complex. The protein gets dropped off into the Golgi complex. Sometimes some things are added to it or changed a little bit. And then proteins get sorted and shipped off. So just like how they left the rough endoplasmic reticulum, the Golgi apparatus membrane will fold around the protein and will send them off to where they need to go from there. The proteins now, when they leave the Golgi apparatus, they are ready, they are finally fully assembled, and they're ready to leave the cell or go wherever they need to go. So the sac that was formed here from the Golgi apparatus membrane will fuse with your cell membrane or your plasma membrane. So that's that outside layer of the cell. So it will fuse with that outside layer of the cell. It will open up, and then it will eject that protein out of the cell and into your body. Let's do a quick review question here. The cell's nucleus holds the cell's primary complement of blank, which contains information for the production of blank. So the cell holds the primary complement of DNA, which contains information for the production of proteins. So that's one of the most important things that happens in your cell, is that DNA being read and producing proteins, and then that controls everything else that goes on everywhere else in your body. Now you've learned all about how proteins are produced in the cell, one of the most important jobs of your cell. But there are also some other things that go on inside your cell outside of protein production that are important. And so we're going to go through those parts next in the cell that don't relate to protein production. We've talked a lot about the rough endoplasmic reticulum, and again, we call it the rough endoplasmic reticulum because it has ribosomes on it that make it look bumpy. There's also something called the smooth endoplasmic reticulum, or SER, which produces lipids. Again, those are fats, and also detoxifies harmful substances that end up in the cell. The type of fat that the smooth endoplasmic reticulum produces depends on the location of the cell. Typical body fat is produced in the smooth endoplasmic reticulum of liver cells and fat cells. Steroid lipids like cholesterol, estrogen, and testosterone are produced in the smooth endoplasmic reticulum of the ovaries and the testes. And the detoxification of harmful substances such as alcohol takes place mainly in the smooth endoplasmic reticulum of liver cells. So let's look at the smooth endoplasmic reticulum specifically in your liver. And we know that the smooth endoplasmic reticulum in your liver helps remove toxins such as alcohol from your body. But the smooth endoplasmic reticulum is only, just like any machine processing anything, it can only go at a certain speed and remove a certain amount of alcohol over a certain amount of time. The excess alcohol that can't be removed can destroy or change your liver cells in 3 different ways. One is that fat can collect in your liver cells, so it can't process all that alcohol, and so you get a fat buildup of fat cells in your liver. This is called fatty liver. This can sometimes be reversible if you were to stop drinking the large amount of alcohol, and that could slowly get better over time. The next 2 categories here, damage that can happen to your liver from excess alcohol, cannot be reversed. So when you get too much inflammation in your liver due to excess alcohol that can't be removed by the smooth endoplasmic reticulum, you can get inflammation. This is called alcoholic hepatitis. And you can also get permanent scarring of your liver called cirrhosis. And a severely scarred liver can lead to liver cancer or death. Alcohol that's taken with medications, both over the counter and prescription, can be especially dangerous because these medicines can interfere with the breakdown of alcohol. So again, your smooth endoplasmic reticulum is like this machine that's meant to break down toxins such as alcohol in your body. And medications can impact how that machine works. And so they can interfere with the breakdown of the alcohol and make that liver damage even more severe. So you should never take alcohol and medications together, especially medicines that have acetaminophen like Tylenol. So we don't think of Tylenol as being very dangerous, but too much Tylenol, even by itself, can damage your liver, but Tylenol, especially with alcohol, can damage your liver. And this is from the Toronto Center for Liver Disease. They give you a list of just some medicines that you should not drink with alcohol that are dangerous to have with alcohol. And these include antibiotics, blood thinners, antidepressants, sedatives, pain medications, and muscle relaxants. So these all influence the way the alcohol gets broken down and the effect of the alcohol on your body. According to the Toronto Center for Liver Disease, women with a healthy liver should not have more than 1 alcoholic drink per day, and men with a healthy liver should not have more than 2 alcoholic drinks per day. But individuals who have liver damage should not drink any alcohol because no amount of alcohol is safe for individuals with liver damage. And this is because your liver is damaged, and therefore that smooth endoplasmic reticulum is not functioning as it normally should be, and you're not able to remove that toxin from your body, leading to even more severe and increased damage. You won't feel anything until about 75% of your liver has been damaged. So you would not notice anything different or know anything's wrong with you until that much of your liver has been damaged. And by then it may be too late to do anything about it. So what you can do is see your doctor and be very honest about how much alcohol you drink. And that will allow your doctor to assess if you need any blood tests to help your doctor find early signs of liver damage. So if you're drinking a certain amount of alcohol, it would be good to get your liver damage detected early. And again, that can be done by blood tests with your doctor to find those signs early before you reach this 75% damage where you start feeling symptoms and then maybe can't do anything about it. Let's look at, so we've talked about alcohol in general, but alcohol in young adults is especially problematic. And when I talk about the young adult group, I'm talking about the young adult brain, and this is about 18 to 24 years old. And this is where your brain is still developing neurobiologically into the full adult brain that it will become. And so, according to society, you are technically an adult at whether you look at 18 years in general or 21 years for alcohol. But anyone in this category, your brain is still different than an adult brain. It is still growing, and alcohol affects your brain in this age category differently. So, and this is for a few reasons. So first of all, the brain in this age group, or even younger, so when I talk about this age group, younger than this age group would fall into the same category as this. So the brain in this age group is designed to be curious about the world and want to learn about it. And while parts of this are good because it's great for you to want to go learn about the world and learn things that are useful to you in the rest of your life, this type of brain connection at this point leads to more risk-taking behavior and less concern about those risks. And so people consuming alcohol during this age group are less likely to think about the effects of the alcohol on them and be likely to consume more alcohol than is safe. The second reason that alcohol in this age group can be a problem is that the young adult's brain is actually less sensitive to alcohol sedation. So alcohol has a suppressive effect on your system, and which means it slows things down, makes you more sleepy, and you won't feel the effects of this until more damage is done. So normally you'd reach a certain amount of alcohol that your body would sort of start slowing down, and you'd realize that you're feeling the effects of that. The young adult brain does not feel those effects until more alcohol is consumed, and so in a sense you don't feel the effects until more damage is done to your liver and your brain. And finally, this age group is more sensitive to alcohol-related disruptions in memory. So your brain is more easily damaged by alcohol in this age group. And so essentially the big takeaway from this is even though alcohol is legal at 21 years old, your brain is still developing, and it would be good, if nothing else, to think about this and be aware of that when you consume alcohol during this age range, that it does pose a greater risk to someone who is older who is drinking alcohol. And so alcohol tends to seem very cool at this age, I think partly because of that increased risk-taking behavior. And so alcohol is often seen as a cool thing, but drinking too much alcohol during this age group and having permanent liver and brain damage is definitely not a cool thing to have to live with for the rest of your life. Any factory would need a way to get rid of waste, and in the animal cell, the organelle that does this job of getting rid of waste is called a lysosome. It's this word here. And lysosomes not only get rid of old waste, they can break down material to recycle it, to be reused in the cell, and they can process new material before it's ready for use. And that's all done in the lysosome. There can be several hundred lysosomes in each cell, and lysosomes are like sealed off acid vats that take things in. They use this acid and enzymes to break things down into small pieces, and they can return these small pieces, if there's something the cell can reuse, to the cytosol, that fluid within the cell. And what is bad for the cell or doesn't need to be reused will either be stored inside the lysosomes, so it can store as like a garbage can for your cell, or the lysosomes can export these sacks of waste outside of the cell so that the rest of your body can remove that waste from your body. They can also work on breaking down things that are trying to enter the cell, like a pathogenic bacteria. And so if it senses something bad, some kind of bad bacteria trying to get into your cell, it can send lysosomes to the edge of the cell and try to break down that bacteria before it enters and damages your cell. A little bit more on lysosomes. The rate at which lysosomes work depends largely on a person's nutritional state. So again, lysosomes help break down pieces in the cell to be reused if need be. And so if you were starving, lysosomes will break down perfectly functional organelles and proteins at a really fast rate to meet your immediate energy needs. And so it'll take parts of a cell that are working fine, but it says I'm starving and I need energy now and I'm not getting it from food. So I'm going to break down my own cells to get that immediate energy need. This would be like throwing furniture into the fireplace to keep warm. You do it as sort of a desperate measure to survive. It wouldn't be your first option, but if you were freezing cold and that was your last option to survive, that might be, your furniture wouldn't be so important anymore. So this is done without regard to the long-term consequence, but saying that I need this energy right now just to survive. In a person who's well fed, the cellular breakdown and recycling occurs much more slowly and only when parts wear out. So they don't normally break down good cell parts for energy unless you're starving. So it may sound good to just be completely well fed and never have cellular breakdown, but the flip side of this is that underactive recycling of old materials. So if your cells are not getting rid of old materials quick enough, it can lead to Alzheimer's disease. And this is due to faulty protein fragments that are allowed to build up in your brain cells rather than being broken down. And then the brain cells die because of the proteins that have accumulated in them. So you really need a balance between your cells breaking down things, having energy and breaking them down at a good rate and not destroying themselves, but not breaking down things so slowly that waste builds up and your cells die and you get something like Alzheimer's disease. Next, let's look at mitochondria. This is another organelle within the animal cell, and everything that your cells do takes energy, and this energy comes from food that you take into your body, and then each of your cells take in this food into an organelle called the mitochondria, and that's this organelle in these pictures here. And it's sort of like a series of folded membranes. You can see the actual microscope picture here of what it looks like. And the mitochondria take in food, they break it down and turn your food into a form of energy called ATP, which stands for adenosine triphosphate. And this is like the energy of your body, the energy form that exists inside your body. In order to produce ATP, this energy source, mitochondria need oxygen. And that's why you need to breathe. Your cells are constantly needing a supply of oxygen, or they can't break down your food into energy. And if your cells can't break down food into energy, they can't do anything without that energy. So that's why you constantly need to breathe, and your cells are constantly also needing a food source. And we'll go over this in a lot more detail in chapter 7 on the process called respiration, which is taking in that oxygen and breaking down food for energy. Okay, the last structure that we'll cover in the animal cell are microfilaments. And the cytoskeleton, or sort of like the skeleton that exists within your cell, is made up of a network of protein filaments. So these fibers, sort of like rods made of protein that give the cell structure, they help with movement, and they aid in the transportation of materials within the cell. And there's a few different types of structures that make up the cytoskeleton. They're listed below and on the right from smallest to largest. So, and these are microfilaments, which are the smallest, intermediate filaments, which are, you could probably guess, intermediate or medium in size, and microtubules, which are the largest type of filament within the cytoskeleton. So let's go over first. Microfilaments are the smallest protein fibers that support the cell or give it structure, and they help move and capture prey. So microfilaments help a cell move. The way that they do this is pretty cool. They break down on one end really quickly, and then they grow really rapidly on the other end really quickly. So they can almost shoot off like an arm, grab prey, bring it back in, and that's by growing these microfilaments really quick on one end and then pulling them back in. So they move by building up and breaking down, and it's pretty cool, all the things that your cells can do. Next is intermediate filaments, and these form a more permanent form of skeletal support system for the cell. So it would be somewhat like the skeleton or the bones inside your body. Their main function is to maintain the cell shape. Lastly, we have the microtubules, or the largest filaments in the cell, that they help give the cell its overall shape, and they also act like a roadway transporting structures from one part of the cell to another. So we've talked about how all these different things like ribosomes and things move around your cell, and this happens on this sort of roadway of microtubules that exist within your cells. There's two types of microtubular extensions that can exist off the cell. So we talked about microtubules inside the cell, but there's a few that exist on the outside of the cell, and two of these are cilia here, which are very short little hair-like projections, and then flagella, which are long tail-like extensions. And cilia are, as I said, the short hair-like growths off the cell. They're made of microtubular extensions, so these microtubules or filaments that come off the cell. And some examples of these in your body are, we have a lot of cilia lining our nose. They help move foreign particles out of your nose. So if you have things like dust that come into your nose, they help move that dust out. And they also help transport things into your nose, like smells, to take that smell signal into your nose and process that so you can smell things. Cilia also line your lungs to help clean out foreign material that has been inhaled. They act sort of like a broom just sweeping, that would sweep dirt off the floor. They help sweep foreign particles like dust out of your lungs. And our other type of microtubular extension are flagella. And again, these are long tail-like extensions. They often function in cell movement. So they act sort of like a tail, helping things typically like single-celled animals or bacteria to swim along. Let's move on now to the plant cell. And the plant cell has a lot in common with the animal cell, which we won't go over again. So it has all of the parts on this list here that exist in both a plant and animal cell and do the same job, which are the nucleus, still containing that DNA and producing that RNA that's taken out of the nucleus to produce proteins, still has smooth and rough ER and ribosomes to help aid in the production of proteins. There's a cytoskeleton that helps give the cells support and transport materials through the cell. The cytosol, or that liquid that's contained within the cell, contains nutrients and moves things through the cell. There is the mitochondria that exist in both the plant and the animal cell for energy, and there is a plasma membrane within the plant and animal cell that can control what goes in and out of the cell. Some things that are different in the plant cell compared to an animal cell is it has something called a central vacuole. It's this large water sac here within the plant cell, and this is important for one, that it stores water inside the plant cell, it stores nutrients in the plant cell, it can help maintain cell pressure by filling up with water and maintaining that pressure within the cell. If you've ever seen a wilted plant, that's because it's lost water in that vacuole and it doesn't have that structure anymore, that support, so it flops over. And the central vacuole also manages cell waste like the lysosome of the animal cell would. So plant cells don't have a lysosome, but they do have the central vacuole that can do the same thing. Plant cells also have a cell wall, which is this rigid structure around the plant cell. If you remember, animal cells just have a cell membrane. It's bendable, and so you get a round-shaped cell, and it can bend and change in shape. The plant cell, however, that wall is rigid, and it gives these plant cells a very rectangular shape if you look at them under a microscope. Plant cells also have a cell membrane inside the cell wall, but it's much thicker. The cell wall is much thicker than the cell membrane. So cell walls, what they do for the plant is they give it strength, they restrict the amount of water a plant cell can absorb, so it can't just absorb so much water it explodes, and they protect the cell from harmful outside influences, so they keep bad things from getting in. So if they're so useful, you might wonder why animal cells don't have cell walls, and cell walls are great for making the plant rigid, and it's great for something that needs to stay in one place. Think of a big giant tree. It's very strong, it's very sturdy, but it doesn't have to move. Animals, on the other hand, need to be mobile. They need to catch prey and avoid becoming prey, and the flexible cell membranes helps allow them this mobility. It helps them be able to move around much more easily. Okay, our last part that we'll go over in a plant cell that's different from an animal cell is the chloroplast. And a chloroplast is where something called photosynthesis occurs in a plant, and that's these little green things here. And photosynthesis is where a plant cell takes in energy from the sun along with carbon dioxide from the air and some water, and it turns it into sugar to be used as energy. So they just sit there in the sun, they don't eat food, they just make their own food by using energy from the sun. Wouldn't it be really nice if we could get all our food just sitting around in the sun? But we can't do that. We have to actually ingest our food into our body and not make our own food. But plants are pretty unique in that they can make their own food source, and we'll go over a lot more on this in Chapter 8. If you are looking for a comparison and want to go over a good review of parts in the plant and animal cell, if you look in Table 4.1, I think it should be on page 82 in your text, there's a good summary of all the structures in plant and animal cells and what they do. We're going to skip the section on cell-to-cell communication. You can skip this in your outline as well, but do keep in mind that cells, whether they're plant or animal cells, are constantly in communication with each other, passing material back and forth in order to communicate with each other. That's the end of our Chapter 4 lecture, so I will be in touch with you again next week. I hope you have a great week, and let me know if you have any questions as you go through your work this week.